Hi everyone, welcome back to my A-level biology lecture series. In the previous lesson, we have explored the overview of the meiosis, meiosis 1, as well as meiosis 2. In this lesson, we will explore the differences between mitosis and meiosis. Let's get started. The diagram summarizes the differences between mitosis and meiosis. One of the differences we can see from the diagram here, mitosis produces two genetically identical cells. However, meiosis produces four genetically unique daughter cells after two rounds of meiosis, which are meiosis 1 and meiosis 2. However, mitosis only takes place one cycle to produce two genetically identical daughter cells. So, what are the other differences between mitosis and meiosis? We can look at the type of parent cells where mitosis occur in both diploid and haploid cells. Meiosis can only occur in diploid cells only. To complete the mitosis, there is one cell division. However, to complete the meiosis, there are two cell divisions occur, or two nuclear division more precisely, which are meiosis 1 and meiosis 2. A total of two daughter cells are produced after mitosis. However, four daughter cells are produced at the end of the meiosis. In mitosis, these two daughter cells are genetically identical to the parental cells as well as between these two daughter cells. However, in meiosis, these four daughter cells are genetically different or genetically unique compared to the parental cells, as well as among these four daughter cells. At the end of the mitosis, every single daughter cells have the same number of chromosomes as the parent cells. If the parent cell is a diploid cells, then the daughter cells are diploid. If the parental cells are haploid cells, then the daughter cells are haploid. For meiosis, we can see that the number of chromosomes is half as compared to the diploid parental cells. During mitosis, homologous chromosome pairing does not occur. How about meiosis? Yes, homologous chromosome pairing occurs during prophase 1 to form bivalents or tetrads. Crossing over does not occur during mitosis. However, crossing over occurs during prophase 1 when this crossing over take place between these non-sister chromatids where the allele can be changed. These introduce genetic variations. Independent assortment of homologous chromosome does not take place during mitosis. However, independent assortment take place during metaphase 1 as well as metaphase 2. Metaphase 1, homologous chromosome align at the metaphase plate. Therefore, independent assortments of homologous chromosome take place during metaphase 1. And we do know that due to the crossing over, sister chromatids are no longer identical. So therefore, the way how they align at the metaphase plate during metaphase 2 creates the or introduce the variation. So in this metaphase 2, independent assortments of sister chromatids occurs. What are the functions of mitosis in organism? So generally involved in growth, tissue repair, and as well as asexual reproduction. And another one that we can actually include is during immune responses, where clonal expansion take place during mitosis. For meiosis, the function generally is to produce the gametes, either male gametes, sperm cells, or female gametes for sexual reproduction. As well as productions of gamete, meiosis introduces genetic variations. And with that, we have come to the end of this lesson. Hope you found it helpful and easy to follow. If you have any question, feel free to drop them in the comment section. See you in the next lesson and have a great day.